Hello everyone! Today we're going to be talking about Staphylococcus aureus, or S. aureus, and its antibiotic-resistant form, MRSA, short for methicillin-resistant Staphylococcus aureus. These bacteria are important because they're common and pose serious health threats in hospitals, schools, and everyday environments. Staph infections range from minor skin conditions to life-threatening issues like pneumonia or bloodstream infections. MRSA, in particular, is a global concern due to its resistance to antibiotics, making infections harder and more expensive to treat. Understanding the biology behind both is key to stopping their spread. Staphylococcus aureus is a gram-positive bacterium that forms clusters of round cells resembling bunches of grapes, which is where the name Staphylococcus comes from. It's commonly found on the skin and in the nose and throat. MRSA is a strain of S. aureus that has developed resistance to methicillin and similar antibiotics. Methicillin was once effective against S. aureus, but MRSA can survive despite its presence. MRSA infections are often a concern in healthcare settings, but are also spreading in the broader community. Both S. aureus and MRSA are gram-positive cocci, meaning they appear purple under a microscope due to their thick cell wall made of peptidoglycan. They share surface proteins that help them adhere to host tissues and enzymes that allow them to invade and evade the immune system. The main difference is that MRSA has a genetic adaptation that allows it to resist methicillin. The genomes of S. aureus and MRSA are similar, but MRSA has acquired the SCC-MEC element, which contains the MEC-A gene. This gene encodes PBP2A, a modified penicillin-binding protein with low affinity for methicillin and other beta-lactam antibiotics. This allows MRSA to continue synthesizing its cell wall even in the presence of antibiotics. Penicillin works by binding to penicillin-binding proteins in bacteria, which are crucial for building the cell wall. When penicillin binds, it prevents the bacteria from forming a strong cell wall, leading to cell death. In S. aureus, penicillin is effective because the bacteria rely on PBPs to build their cell wall. However, MRSA has a mutated version of these proteins. The PBP2A, produced by MEC-A gene, has a low affinity for beta-lactam antibiotics. This is similar to a lock and key mechanism. The antibiotic is the key, but the lock, which is the PBP2A, has been altered so that the key doesn't fit properly. This allows MRSA to continue building its cell wall, making it resistant to methicillin and similar antibiotics. In conclusion, S. aureus and MRSA are similar in structure, but differ mainly in their ability to resist antibiotics. MRSA's resistance to methicillin makes it a major health concern, especially in hospitals. However, the rise of MRSA has pushed researchers to find alternative treatments like bacteriophages, viruses that target and kill bacteria, and antimicrobial peptides, which are natural proteins that destroy bacteria. Vaccines against S. aureus are also being developed to prevent infections and reduce the need for antibiotics. Beyond new treatments, antibiotic stewardship, using antibiotics responsibly, is crucial to slowing down the development of resistance. Efforts to reduce unnecessary antibiotic use on a global scale are key to fighting the growing threat of antibiotic resistance. By studying MRSA's resistance mechanisms, we can better understand how to fight these infections. This video should give you a stronger foundation for the MRSA case study we explored earlier this semester and encourage you to think about how we can stay ahead in the fight against resistant bacteria. Thank you for watching.